Hello dear viewers, myself Nasir and you're watching Mr.Zoologist YouTube channel. Today we are going to discuss about animal kingdom. What is an animal? An animal in actual are multicellular, either unicellular or colonial. Animals are in most of their forms are multicellular and heterotrophic organisms without cell wall and chlorophyll. Animals lack cell wall, but some of them might have a cell wall like bacteria. So animals are heterotrophic in mode of nutrition, means to say they cannot prepare their own food and they lack cell wall because animal bodies contain cell membrane. Some of the exceptions are also present, like fungi have a cell wall and algae have a cell wall, bacteria have a cell wall. 11 major phyla of animal kingdom are phylum Porifera, phylum Nidaria, phylum Ctenophora, phylum Platyhelminths, phylum Aschelminth, phylum Annelida, phylum Arthropoda, phylum Mollusca, phylum Echinodermata, phylum Hemichordata, phylum Chordata. These are 11 phylums of the Kingdom Animalia. Some smaller phylums are also present. What are bases of classification? On which basis do we classify animals? First of all, levels of organization, how much complex body form is. And next is body symmetry. Either body symmetry is radial or bilateral. On this basis we classify the organism. Next is germinal layers, means to say mesoderm, endoderm, ectoderm. On the basis of these layers we classify organisms into triploblast and diploblast. Fourth is coelom or body cavity, means to say which form of body cavity is present. Fifth, on the basis of metamerism, means to say how many if segmentations are present and if segments are absent. Next is classification basis notochord. Notochord develops in some organisms or in the larval stages or in the embryo stage, which functions as a brain, primitive brain. Basis of classification, in which first of all we are going to discuss about levels of organization. Based on organization of cells, animals grouped into 4 levels: tissue level organization, organ level organization, and organ system level of organization. In cellular level organization, small atoms combine together to form cells, and similar cells performing the same function form a tissue, and similar tissues performing the same function form a organ system. Organ level, different organs performing same function form organ system for organization. Cellular level organization, cells are arranged as loose cell aggregates like porifera, whereas cells are arranged into tissues in cnidarians and ctenophoras, and whereas tissues are arranged into organs in higher animals like platyhelminths and chordates. And next, organs are associated to the organ system. Each system performs a physiological function in higher animals, invertebrates of higher levels or of higher organization. Organ systems of different animals show complexities, like digestive system is of 2 types: incomplete and complete. Incomplete has only single opening, that is called a sac-like digestive system, like in Hydra and in Planaria. Whereas complete has 2 openings, mouth and anus. From one food enters and from one food leaves. From mouth food enters after digestion in the stomach, and in the digestive system if the waste is released from the anus. Complete digestive system is seen in roundworm and higher vertebrates. Circulatory system is of 2 types, open and closed. In open, no blood vessels are present, simply blood pumps and circulates directly blood into the body. Whereas in closed circulatory system there are blood vessels, capillaries, arteries, veins, etc., which are involved in the blood circulation. Next is symmetry. It is arrangement of similar body parts on 2 sides of main axis of the body. Based on symmetry, animals are of 2 types: asymmetrical. Body cannot be divided into 2 similar parts, most like perforans, snails. They do not have a symmetrical body, they have asymmetrical body plan. We cannot divide them into 2 equal parts, whereas symmetrical body can be divided into 2 similar parts like planaria. On symmetry, we can also divide it with the help of radial symmetry and bilateral symmetry. What is radial symmetry? Here body can be divided into 2 equal halves by any vertical plane along central axis, means to say oral axis. From oral cavity to anus, we can divide it into 2 equal halves. Some poriferans, cnidarians, ctenophores, and echinoderms adults also possess a radial symmetry, whereas young ones have bilateral symmetry of echinoderms. Next is bilateral symmetry. Here body can be divided into right and left halves in 2 equal planes into two equal halves in only one plane. We can divide it in one plane, whereas in radial symmetry we can divide it into multiple planes. For example, platy elements to chordata except adult echinodermata all have a bilateral symmetry. We can divide them in only one plane, not in more than one plane. Here body cavity or body symmetry can be divided into equal halves by any plane. We can cut down in the radial symmetry an animal along any plane. For example, some poriferans and cnidarians, for the hair body can be divided into right and left halves in only one plane. For example, platyhelminths to chordata. In some other animals, we are looking just for the sake of example. The body of bilaterally symmetrical animals has a dorsal side, upper side, a ventral side, lower side, left and right lateral sides, anterior side cephalic, posterior anal or tail side. That are the portions for the organism to be described with the help of the symmetry. How much does it looks like, or what type of it is, radial or bilateral. Next, we are talking about germinal layers. These layers are formed from the embryo, which are all the body organs formed. From these layers, all the cells, all the organs are formed. Based on the number of germ layers, animals are of two types: diploblastic animals and triploblastic animals. Diploblastic animals, two germ layers, outer ectoderm and inner inoderm is only present. Mesoderm is absent. In between these layers, an undifferentiated jelly-like layer called mesoglea is present, like cnidarian and ctenophorus. Three germ layers are formed in triploblastic animals: ectoderm, outer, middle mesoderm and inner endoderm. For example, platyhelminths to chordata all possess triploblastic organization. Next is coelom or body cavity. It is lined by mesoderm. It is seen between body wall and gut wall, means to say between external wall to the cytoplasmic inner extent is present. Coelom. This cavity or coelom, if it is present in gut, then it is called gut cavity. Coelom separates the muscles of gut and body wall. Based on the animals are of three types. Nature of animals: acoelomates, in which no body cavity is present, and second is pseudocoelomates, in which false coelom or false cavity is present, and coelomates, in which true cavity is present. We are going to discuss it in detail now. Acoelomate, space between body wall and digestive cavity is filled with matrix parenchyma. For example, porifera to platyhelminths, there is no actual cavity, whereas in pseudocoelom, here body cavity is not lined by mesoderm. Mesoderm is scattered pouches. For example, Acoeloms, in which they have false cavity or false coelom. Whereas in coelomates, coelom arises from the mesoderm. Coelom is lined by peritoneal layer and filled with coelomic fluid. For example, Annelida to chordata all possess coelomic cavity. We are going to elaborate it more in the detail. Space between body wall and digestive cavity is filled with matrix, like porifera to platyhelminths. You can look at it in the diagram. There is no cavity between. Here the body, whereas in pseudocoelomates, body cavity is not lined by mesoderm again. Scattered pouches are present. Mesoderms, false coelom or cavity, you can look at in the diagram in the whited area shown, and the red area is muscle layer. Third one is coelomate. Here coelom arises from mesoderm. You can look at in the diagram that coelom small pouches are arising that are in actual present.